Good morning and hi everyone. I am Hamida Ghani, a bioinformatician at novoCraft Technology. Thank you for attending our live webinar on NGS analysis platform for SARS-CoV-2. It is my pleasure today to introduce you with our latest tool for SARS-CoV-2 NGS viral analysis using novoWorx platform. Before we start, I want to highlight that our viral NGS application is for research use only and is not suitable for diagnostic purposes. At the end of the webinar, we hope that the attendee will be able to perform bioinformatic analysis and standardize best practices in viral analysis using novoWorx, and also to apply novoWorx viral analysis pipelines of SARS-CoV-2 screening and classification analysis for SARS-CoV-2 virus detection and SARS-CoV-2 reference assembly analysis using either virus or virus plus human as their reference genome. We have three main sessions. In the first session, I will provide a brief introduction on novoCraft and the pipeline that we are going to discuss today. The second session by Farhan Tahe, novoCraft bioinformatic programmer, will cover the theory and also a demonstration of SARS-CoV-2 screening and classification analysis. The third session on SARS-CoV-2 reference assembly analysis will be covered by Chong Su-wei, our bioinformatic programmer. We are going to have a small session for Q&A at the end of the webinar. Attendee may ask questions using our chat system. Please submit your question in the chat box throughout the webinar and we will address it during the Q&A. If we are unable to address your question during this event due to time constraint, we will email the answer to you. To those who are new to novoCraft Technology, here is a little bit of background for our company. We are a group of bioinformatics software developers and solution providers that are committed to empower research through developing the software and platform for our client. novoCraft has been in operation since 2008 and we are based in Petaling Jaya, Selangor, Malaysia. We offer a broad portfolio of bioinformatic analysis and solutions, including proprietary bioinformatic tools, services, consultancy, and training. Currently, we have more than 200 licenses globally, and in the last decade, we have a consultancy track record spanning across several sectors such as pharmaceutical, medical research, and agrobiotechnology. Now, back to the main topic of this webinar. The emergence of SARS-CoV-2 has caused the current pandemic with the disease known as COVID-19. The pandemic has caused a global health virus threat, resulting in millions of confirmed cases and deaths worldwide. Genomic analysis is one of the important approaches in understanding the origin, transmission, and the control of the new virus. Insight from the virus genetic will allow the prediction of longer-term, larger-scale trend and independent validation in estimating the epidemic growth. In addition, identifying unique genetic changes shared by all those infected in a single virus transmission chain will distinguish between clusters and add to the information provided by patient contact tracing. Next, genomic analysis will also help provide important information on clinical significance of different virus strains. Certain mutations may alter how the immune system recognizes viruses resistant to antiviral drugs and also provide important leads to vaccine development. Our tools have been used in previous virus outbreaks such as Zika virus and also Ebola virus using novoWorx to perform the disease surveillance and transmission study. The outcome of this study include the reconstruction of viral transmission and also SNP identification. novoWorx is our flagship alignment software that is compatible with various major short-read NGS platforms. It can be used to align short-read data from Illumina, MGIC, Iron Torrents, among others. novoWorx has made a significant investment in developing highly precise and accurate tools. novoAlign especially is used as part of standard protocols for several organizations. Here, Broad Institute Viral Genomics Group used novoAlign as part of its standard pipeline for viral genomic analysis as highlighted in the figure. Next, looking at the COVID-19 pandemic timeline, the COVID-19 pandemic started at the end of December 2019 in Wuhan, China, and spread rapidly worldwide. By January this year, the first whole genome sequence of SARS-CoV-2 was published, and many initiatives have been developed to support COVID-19 outbreak responses using genomics, including NextStrain and GSAID. In April 2020, the complete genome sequence of SARS-CoV-2 in Malaysia was deposited in the NCBI database. Taking this into account, we have developed a SARS-CoV-2 viral NGS analysis solution to help researchers reach their goal by studying the virus outbreak. Okay, we are looking on the next slide. Many countries have come up with genomic study using NGS to perform research on samples from COVID-19 patients. However, there are few challenges to overcome. One of the challenges is in handling the large number of samples and the amount of output data from the sequencing process. Automation in analysis workflow would really help in this case. Not only would it reduce human error from manual processes, a good automated workflow will also be more time and cost effective. It is also important to choose the right tools that are capable of producing high quality draft genome, especially during the reassembly process. Additionally, users may need a predictive analytic part for virus screening, classification analysis, and risk coverage plot. The most challenging part, of course, is the concern about the result outputs management. It is imperative to choose tools that produce user-friendly results and make it easier for the researcher to manage the output and interpreting data. So we developed the solution, which is a SARS-CoV-2 NGS pipeline powered by novoWorx platform. novoWorx is an easy-to-use platform that is already in the market for a couple of years. This platform provides a number of flexibility across different NGS studies. For SARS-CoV-2 analysis, the first pipeline is for virus screening and classification analysis. The second pipeline is for the virus reference assembly. User can choose to use virus or human plus virus as the reference genome for the assembly process. Another pipeline that is currently being optimized is the virus variant calling and annotation pipeline will be made available soon in novoWorx. User can run the bioinformatic analysis without having to deal with complex algorithms with this automated analysis. In general, if users have raw dataset in FASTQ format obtained from the sample sequencing output, user can perform the pre-quality check to see the quality of the raw dataset and followed by the virus screening process using virus screening and classification pipeline. This is an effective and high throughput detection of the SARS-CoV-2 virus for the unbiased pathogen discovery. User can then view the result as in the table or corner chart figure. Our SARS-CoV-2 pipeline can accurately detect the virus in the sample dataset. Next, for the screening pipeline, the SARS-CoV-2 bioinformatic resources are retrieved from Genexa website using human and virus index based on NCBI RefSeq and NCBI virus genome of version 2. It provides fast viral detection and classification of SARS-CoV-2 and other possible viruses directly from sequence read by using 138 SARS-CoV-2 complete genome. This pipeline will perform comparative analysis based on abundant determination of viruses and contamination of human DNA. Our viral pipeline can provide a QC report to spot any problem with NGS read quality. It ranges from the basic read statistic up to the adapted content detection. User can click the HTML report based on the module output and perform quality check for the given dataset before they proceed with the virus screening and classification. Our pipelines are reusable in which user can also download the QC report for the dataset in a zip file format for their future reference. The virus screening pipeline in novoWorx can give better data visualization using interactive data tables, heat maps, and Sankey flow diagram for the user to analyze the viral classification output. User can click the Open Pavilion button to link the Kraken-like report generated in this pipeline output and upload it using the integrated Pavilion website. An interactive viewer will help in the validation of matches to a particular genome, which is important when using metagenomic experiment for pathogen detection. Next, we are looking into the second pipeline. The second pipeline is for the virus reference assembly analysis. It has two different sub-pipelines, which are viral reference novoLR and combined reference novoLR. User can choose either to use the viral reference novoLR pipeline for virus reference genome only, or combine reference novoLR pipeline if the user wishes to use virus plus human as their reference genome. This pipeline has been optimized for the short-read Illumina and long-read PacBio or nanopore dataset. User can simply upload raw FASTQ sequence file to novoWorx for SARS-CoV-2 reference assembly analysis. At the end, it will generate a final consensus sequence in the FASTA format along with the coverage information. We are planning to track the mutational changes in the virus for the variant calling and annotation pipeline for SARS-CoV-2 study soon. To summarize, SARS-CoV-2 analysis pipeline provides users with seamless workflow across different platforms such as Mac, Windows, and Linux. In addition, the virus pipeline is also equipped with a pre-built virus and human database without the need of downloading and or re-uploading of the database for virus detection. Another features include the various predictive analytics to help researchers with better accuracy and specificity. Interestingly, the pipeline also includes reproducible and reusable output as in the figure and table format. I will end my session here and I would like to pass the next session to Farhan Tahi, who will speak about SARS-CoV-2 screening and classification pipeline. Thank you.
Hi, good afternoon everyone. I am Farhan Tahi, Bioinformatics Programmer in novoCraft Technologies. I will continue our session on SARS-CoV-2 screening and classification pipeline in the novoWorx. Before I go further, I would like to touch a little bit on the basics of novoWorx, which is the platform that we run this pipeline. For your information, minimum requirements to run novoWorx are the PC should be at least have 16 GB RAM memory, 4 physical core with a total of 8 multi-threads, and 1 TB storage hard drive. It's depending on the total size of your input and output generated. novoWorx consists of 4 main sections, which are Data Hub. Data Hub is a place where all the sequence FASTQ files are uploaded into the novoWorx. The second one is references. References is the place where all genome references are uploaded into the novoWorx. The third one is protocol. Protocol is the section where it lists out all the modules and pipelines that are installed in the novoWorx. Last but not least is a project. In project section, user can create and run their analysis projects. That's pretty much of the basics of novoWorx system. Now, let's proceed to the definition of important terms related to this pipeline. Screening and classification. Screening and classification is the process of classifying the abundance of organisms inside the sample, such as human, virus, microbes, archaea, and protozoa. The classification result depends on the database we use in that process. For example, if our database consists of only human and virus genome database, then our classification result will only contain the result from human and virus. Now, we proceed to the input and output for this pipeline. For your information, this pipeline requires input in the form of FASTQ files. So, as long as you have your FASTQ file in your hand, you can run this pipeline successfully. There is no problem. There are four main outputs that are produced by this pipeline. The first one is a corona chart. Basically, it displays the percentage abundance of human and each virus in the samples in the form of corona charts. The second one is heat map abundance table. It will display the percentage abundance and reach count of human and each virus in the samples in the form of heat map table. The third one is Sankey diagram. This diagram will show to us the reach count of human and each virus in the sample in the form of branching diagram. And the last one is a tables. This will portray the overview summary tables for everything that we can be found inside the samples. Now, let's proceed to the workflow of this pipeline. In this pipeline, the FASTQ files from sequencing result or from us will undergo screening and classifying by matching it to the pre-installed database, which consists of human and virus genome database. Once the process is finished, it will generate some visualization output. The first one is corona charts. This chart will visualize the percentage abundance of humans and virus inside our sample. The larger the proportion, the higher the percentage abundance of that specific organisms inside our samples. Next is heat map table. This heat map table will show to us either percentage abundance or the reach count of humans and each virus in the samples. The higher the intensity of the red color, the higher the percentage abundance or reach count of that specific organisms inside the samples. And then we have Sankey diagram. The Sankey diagram will show to us the reach count of human and virus in the samples. The larger the size of the branch, the higher the abundance of that specific organisms inside the samples. Finally, we have summary table. In this summary table, it will show an overall summary of the percentage abundance across the sample. The summary will consist of a number of raw reads in the samples, classified reads, coordinate reads, unclassified reads, microbial reads, and viral reads. Since our database does not consist of any bacteria, fungal, and protozoa, we can't see any percentage in this summary. Let's proceed to result validation. For your information, in order for us to ensure the result integrity for this pipeline, we had carried out result validation by comparing our results to the results that have been done by the others. To do so, we had extracted 7 samples from different projects. The source to the samples are as shown in this table. Among all the 7 samples, 5 of them are classified as detected for SARS-CoV-2. Meanwhile, another 2 samples are classified as not detected SARS-CoV-2. This is based on the result from their respective projects. What we have done is, we have uploaded all 7 datasets into novoWorx and run the screening and classification pipeline. we found out that our results agree with the results from these projects, in which 5 of them are detected, while other 2 are not detected. I would like to show a demo so you can see how easy it is to run this pipeline in novoWorx. Before I start, I would like to introduce you to the source of the dataset used for this demo. I had extracted 2 samples from different projects. The links for those samples are shown in this table. One of them is expected to be positive SARS-CoV-2, while the other are expected to be negative for SARS-CoV-2. This is based on the results from their respective projects. Now, we will start to a demonstration. In order for us to start the novoWorx, just open a tab on your browser. And key in your username and password. First, we go to the Data Hub section. In order for us to upload our FASTQ file, we just need to simply click the Add button here. Then, browse and choose your FASTQ file that you wish to upload. Once the uploaded process are completed, we just need to close these windows. And we will see that our samples or our FASTQ file will be listed inside this section. Now we can move to the project section. In this section, we are going to create a project. To create a project, we just simply click the Add button here. Once we click, we will see the project creation interface. Users need to insert their project name and project description. For example, I will type my classification project as my project name. And I will just copy the project name and paste it to the project description. Next, we move to the next section, which is choose a protocol. In our cases, the protocol that we will be used is a SARS-CoV-2 screening and classification pipeline version 1.0A. If users want to change some parameter for this pipeline, users can simply click Show Settings here. And it will open up the list of parameters. User can change the parameter depending on their analysis interest. If you would like to use the default setting, just ignore these sections. Then proceed to the step of select a dataset. In order for us to select a dataset, we need to mark the dataset that we want to include it in this project. For example, this is the dataset that I want to include, and drag and drop it to the right-hand side of the novoWorx. Then just click Save and Run. Once you click the button Save and Run, what you need to do is just wait for the novoWorx to complete the project. Now, we are going to see the results output generated by novoWorx. For your information, I already ran one project for that purpose. I will go to the projects. Once we see the project status is finished, we can check the results for this project. To do so, we just need to simply click on the Pipeline Output tab here. And it will list out all the output from this project. To view Corona chart, you can just easily click the on HTML file from any samples that you want to view. For example, I would like to view the Corona chart from sample 168. 168 refers to the last 3 numbers from the sample's name. So I will just click the HTML here, and it will lead you directly to the Corona chart. In this Corona chart, we can see that this sample consists about 95% of coronavirus 2, that we can assume that there is COVID-19 inside the sample. And there are around 5% of contaminated human samples inside, contaminated reads inside the samples. But this is for one report for one sample. We also provide a one report that consists of all samples into only one report. To view that corona chart, you go back to the novoWorx, click the project's output, and click the HTML here, and it will lead you to this corona chart. In here, you can see all the samples are listed out here. We can see 770 corona chart. We can see 168 corona chart, and can do a comparison. We can see 168, we have around 95% of coronavirus 2. Meanwhile, in 770, there is no coronavirus 2 detected. We also can see or view a shared summary, which is the shared organism between these two samples. Let's say if we want to export this chart to our journal or research paper, what we can do is just click snapshot here, and you just click download. Then we can export the plot to the journal. That's about the corona chart. Now we proceed to the next output, which is heat map table, Sankey diagram, and also summary tables. To do so, what we can do is just click all and download selected files. Just click yes and continue. Once the download process is finished, we just need to simply click open Pavilion here, and it will lead you to the Pavilion interface. Inside this, what we need to do is just click browse and choose the one that we downloaded before and open. Then it will upload all the samples that already included in this project. We can see there are 168 and 770 already uploaded into Pavilion. Next step is we can choose result overview. In this result overview, we can see the summary tables, what is inside our samples. We can see that in 168, there are 92.1% classified reads, and for 770, there are 88.9% classified reads. We also can see that 7.94% unclassified reads in 168 and 11% of unclassified reads in 770. For your information, unclassified reads are the reads that doesn't map to any human and virus. We also can see there are 79% of virus detected in 168 and 85.5% virus are detected in 770. Yet, we still didn't know on the virus what type of virus, what organism of virus inside the sample. To check about this, we need to proceed to the sample. In this sample tab, we can see that Sankey diagram. We can see the Sankey diagram for 168. We can see that around 34.9K reads of coronavirus presents inside 168. We also can see the Sankey diagram for 770. There are no coronavirus reads inside the 770, but most of the virus are influenza B virus, which consists around 31.5K reads. That's about the Sankey diagram. Now we proceed to the comparison. In this comparison tab, it will show to us the heat map table for the total number of reads or percentage abundance. If you want to see the heat map abundance of number of reads, just let it default. But if you want to see the percentage abundance, just click on percentage abundance and unclick the reads. So you can see the percentage abundance. The higher the intensity of the red color, the higher the abundance of that organism inside the sample. Let's see what is the percentage abundance of coronavirus inside these two samples. Okay. For 168, we can see around 78.96% of coronavirus presents. But for 770, there is 0% of coronavirus detected in 770 samples. But for the influenza B virus, we can see that there are 84.79% of reads are present in the 770, and there is 0% of influenza B virus inside 168. That's all about all the output from this pipeline. Now we will going to the slide presentation. With this demonstration, I will end my session and pass the session to the Chong Sue, who will be talk about novoLR consensus reference assembly pipeline. Thank you.
Good morning, everyone. I'm Sue, a bioinformative programmer at novoCraft Technologies. I will talk about the novoLR consensus reference assembly pipeline built in novoWorx. Before we start on this pipeline, I would like to explain the important basic terms first. Assembly is a term used to describe methods where we combine shorter individual measurements called reads into longer contiguous sequences typically called contigs. In other words, assembly is the process of turning shorter reads into longer contigs. During the sequencing process, it breaks the original DNA or RNA into smaller fragments. So the assembly process is similar to putting together an image puzzle from its many pieces. A consensus sequence is a sequence of DNA, RNA, or protein that represents aligned reads. A consensus reference assembly is a process of turning a consensus sequence is a process of creating a consensus sequence for an individual where the sequence contains variants found in the individual. The novoLR consensus reference assembly pipeline in novoWorx is to create a consensus SARS-CoV-2 sequence for an infected individual. In short, we will take a sample of SARS-CoV-2 RNA patient. The RNA was broken into smaller fragments during the sequencing process, and we will use this pipeline to combine the small fragments into a longer contiguous sequence. There are two sub-pipelines. In the pipeline that uses viral reference, we map reads to only SARS-CoV-2 reference genome. If the sample is contaminated with human DNA, the human reads may be present during the assembly process. So we must make sure our sample contains only SARS-CoV-2 and it is not contaminated with human DNA. If our sample contains human DNA, we will use the pipeline that uses combined reference. We map reads to SARS-CoV-2 and human reference genomes. The aligned human reads will be removed from the assembly process. So what input should we have in order to use this pipeline? First, we need a FASTQ file that contains sample reads from a SARS-CoV-2 patient. Depending on the sequencing method, we obtain one FASTQ file if we have single-end read, and we obtain two FASTQ files if we have pair-end reads. Second, we need a FASTA file that contains the SARS-CoV-2 reference genome. If we want to run the pipeline that uses the combined reference, then we need an extra FASTA file that contains the human reference genome. We have the references provided in novoWorx, so we need to prepare FASTQ input only. The final goal of the pipeline is to generate a consensus SARS-CoV-2 sequence for the patient in FASTA file format. Now I want to demonstrate how to use the pipeline in novoWorx. For demonstration, we will use a subset of the sample taken from the first SARS-CoV-2 patient in Nepal. The patient contacted the virus in Wuhan, China before going back to Nepal. A study on using this sample to generate a consensus sequence has been published, and information about the sample and study can be obtained from NCBI. After demonstration on how to use the pipeline in novoWorx, we will compare the consensus sequence generated by our pipeline with the consensus sequence produced from the study. First, go to the project session. Click on this button to create a new project. For project name, I choose Demo Novo LR. You can choose any project name or description that you want. For the pipeline, I choose this one that uses the virus reference genome only. We have set all the settings for you, so you don't have to worry about this session. Choose this reference. Click on this button to upload any FASTQ or sample file that you have from your PC. Since we have the samples file uploaded, we click this, choose this for our project. Click Save and Run. See how easy we can use this pipeline. Just a few clicks, you can run this whole thing. Now I would like to explain the concept in the pipeline. Our company's aligner software novoAlign maps reads to the SARS-CoV-2 reference genome. It can remove any adapter sequence and low quality base from the reads. After we align the reads, there are regions of the reference genomes where there are no reads aligned to them. In other words, there is zero coverage of reads on the virus genome. If we include the human reference genome in the pipeline, the aligned human reads will be removed from the assembly process. Our company software novoLR Polish combines the aligned reads to generate a consensus SARS-CoV-2 sequence. For the regions where there is zero coverage of reads, the pipeline will put N bases for those regions on the consensus sequence. A coverage plot showing the read depth for each genome position will be generated by the pipeline. The first expected output from the pipeline is the consensus sequence in FASTA file format. We can download the file for our research purpose. Besides, we obtain some output graph. The graph on the left shows the percentage of mapping status of the reads from the sample from the first SARS-CoV-2 patient in Nepal. In this case, we have close to 100% of the total reads, which are uniquely mapped to the reference genome. This means we have a good sample. The graph on the right shows the frequency of fragment reads length for each pair end reads. The graph, this graph shows the percentage of coverage for the sample. In this case, we have close to 100% of the total reads where each of them has at least 30 coverage on the virus genome. The graph on the right shows the read depth for each genome position. We can see that there is high coverage of the reads on most part of the genome. Finally, we compare the consensus sequence generated by our two subpipelines to the reference genome used in the pipeline and also to the consensus sequence produced by the study. We managed to detect the same variants as the one reported by the study that uses viral reference genome only. So this is just to show that our pipeline is produce the same result, the correct result as the study. Okay, that's all from my session. Now we proceed to Q&A session.
Okay, now we are in the Q&A session. If you have any question regarding the virus pipeline or any on the theory or something you are unclear with, please type your question in our chat system on your screen. In the meanwhile, you can also contact our team for this webinar at webinar@novocraft.com. And if you would like to know about the pricing plan for the academic and corporate user, you can always contact our sales team at sales@novocraft.com in the letter. Okay, we are still waiting for the attendee to type and finishing their question there. Yeah, please don't hesitate to ask us a question.
We have one question here. Can this system be used for bacterial amplicon or WGS dataset from any sequencing platform? For that question, actually for that question, yeah, we don't implement any bacterial database in this SARS-CoV-2 screening pipeline. However, users can use other available pipeline for specifically for microbial study only. Yeah, that's my answer.
Okay, I think we have answered the attendee question. And if you have more questions, please don't hesitate to email us later. We will end the webinar session here. We hope you can provide the feedback for this webinar later and have a good day. Thank you.